These are the lecture notes for Chapter 22 on the kingdom fungi. As far as the outline, the sections we're going to cover are evolution and especially the characteristics of fungi. What distinguishes the kingdom fungi from, like, for example, the kingdom Animalia, the kingdom Plantae, and then the diversity of fungi. There are 5, sometimes 6, depending on which textbook that you're reading, fungal phyla. So remember in the last chapter, in chapter 21, we classified the protista into supergroups. The fungi are already in a supergroup called the Opistheconta, along with animals and anoflagellates. But they're additionally, the way that we're going to learn them is we're going to classify them into different phyla. So the phylum is what's going to be important this time, not the supergroup. And then there are some 2 symbiotic relationships that are very important as far as, within ecosystems. So starting with characteristics of fungi in general, make sure that you remember that even though this is the kingdom fungi, we're still within the domain Eukarya. In fact, starting with this chapter, starting with the previous chapter, all of our organisms from here until the end, until maybe the last couple of chapters, are just strictly within the domain Eukarya. So domain Eukarya means that the cells of these organisms have a nucleus and membrane-bound organelles. This supergroup is the Opistheconta, and the kingdom is fungi. So there are 6 phyla that we recognize. We're going to focus primarily on 5 of them. Most fungi are multicellular, but we do have unicellular yeast. Fungi are heterotrophic, meaning they have to consume their food. They don't make their own food. And they're saprotrophs. That means that they are decomposers. They release digestive enzymes outside of their body or outside of their cells. And those enzymes will break down the substrate that they're growing on, the organic molecules within, like, dead plant material or animal material, dead and dying. Anyway, it will allow them to, the digestive enzymes will break down those molecules and allow them to absorb the nutrients back into their cells. So they do their digestion outside of their cells, outside of the fungus, and then they absorb what has been digested. Now, these are the fungal groups, and the ones we're going to focus on are highlighted. The Zygomycota, we're going to focus on primarily the black bread molds. The Chytridiomycota, we call these the chytrids. Probably the most common example of those is one that causes, I'm going to try to say it. I'm going to be brave. Batrachochytrium dendrobatitis, I think is the name of it, but anyway, it is the specific fungus that we're going to learn about. But the chytrids cause a skin disease in amphibians, particularly frogs, and there is one in particular, the one that I just said that I'm not going to try to say again. This one has wiped out frog populations, like all over the world, but especially in Africa in recent years. Then there's the Glomeromycota, and these, the main example within this phylum is going to be the ones that form mycorrhizae, which is a symbiotic association between fungi and plant roots, helps to increase the surface area of the plant root. Then there's Basidiomycota, which are your traditional mushrooms. And the Ascomycota, which is going to be your most diverse group. It's going to include one called Aspergillus, another one called Penicillium. But the Ascomycota as a group are called the sac fungi, whereas the Basidiomycota are the cup fungi. we really won't learn much about the Blastocladiomycota or the Neocalymastigomycota. So it's just going to be Zygomycota, Chytridiomycota, Glomeromycota, Basidiomycota, and Ascomycota. These are the fungal phyla that we will focus on in chapter 22. And then just it is generally believed, and the evidence shows us that probably the common ancestor of fungi and animals was a flagellated unicellular protozoan. Okay, so most fungi are multicellular. The yeast are the unicellular fungi. So yeast like that's used to make bread and beer, and then yeast that cause yeast infections. Those are unicellular fungi. But multicellular fungi is organized into mycelium, like a mycelium, which is just a collection of these thread-like filaments called hyphae. So each individual thread is a hyphae, and the collection of hyphae is called the mycelium. The cell walls of fungi is composed of chitin. If you remember, the cell walls of bacteria are composed of peptidoglycan, but fungal cell walls are composed of chitin. And just like with animals, animal cells store glucose or excess food as glycogen. So do fungal cells. Fungi store excess food as glycogen. So make sure that you know that animal cells and fungal cells, this is one thing they have in common, is that they store glucose as glycogen rather than starch. Plant cells store glucose as starch. All right, let's see. And you can see some hyphae growing on the surface, some fungal hyphae threads growing on the surface of the strawberries. And then this is what they would look like. This is an illustration of what they would look like under a microscope. And they're called septate if they have walls, cross walls, kind of like between nuclei. And then if they don't have cross walls between the nuclei, they're called aseptate. But hyphae are the thread-like structures, and then mycelia is the collection of hyphae. All right, there is both types of reproduction in fungi. There's sexual and asexual. The sexual reproduction involves 3 stages. The hyphae are haploid, as you can see down here, the haploid hyphae. This is a septate hyphae, and you can see the nucleus between each cross wall is haploid, so it only contains 1 set of chromosomes. Then you have a dikaryotic stage where the nuclei within each individual unit of the hyphae, there are actually 2 of them. There are 2 haploid nuclei, and that's what dikaryotic means, 2 haploid nuclei. They haven't fused, they're just in there together. And then you have the zygote, which is when those 2 nuclei fuse and become diploid. And meiosis occurs within those cells and produces the haploid hyphae. So dikaryotic just means paired haploid nuclei. And what it doesn't say here is that when the nuclei fuse and you get a diploid nucleus, the product produces haploid spores by meiosis. So okay, so that's not really shown in this, but when you get this diploid nucleus here, when it divides by meiosis, you get haploid spores that can germinate into a new fungal organism. As far as, okay, so terrestrial or fungi that live on the land usually produce non-motile spores. In other words, they don't swim. A spore is defined as a reproductive cell that develops into a new organism without the need to fuse with another reproductive cell. So a spore is kind of like a seed, but there's not as much to it, Asexual reproduction usually involves the production of wind-blown spores, and they land and they germinate, somewhere. and grow into a new fungal organism. And then unicellular yeast reproduce by budding. They reproduce asexually by budding. Okay, so let's look at the diversity. Let's go through the different groups of fungi, the different phyla. First of all, we already said that the supergroup Opisthokonta includes fungi and animals, as well as the protozoans called choanoflagellates. Now, here is a table that shows you basically the basic characteristics of each phylum that I want you to know. So I want you to know the Zygomycota, and these contain hyphae. They contain aseptate hyphae. That's not as important, but their sexual spores are called zygospores, and the one that you definitely need to know is that the only one that I'm really going to talk about in the phylum Zygomycota is the black bread mold, Then we move on to the Chytridiomycota, or the chytrids. These are aquatic, they are unicellular, and their spores are called zoospores. They can be animal parasites. There's one that we're going to learn about that causes it's a parasitic skin infection on frogs. Then we have the Glomeromycota, which form mycorrhizae. They're called the AM fungi also because they form arbuscular mycorrhizae. Mycorrhizae is a symbiotic association between the Glomeromycota fungus and plant roots. And what happens is the fungus increases the surface area of the plant roots, so the plant roots can absorb more water and minerals from the soil. And most plants do have mycorrhizae in their roots. Then we have the Ascomycota, which is our largest, most diverse group that we're going to learn, phylum. The Ascomycota reproduce asexually using conidiophores and sexually using So the asexual spore is called a conidiospore, and the sexual spore is called an ascospore. Okay? We have a lot of examples within the Ascomycota. They're also called the sac fungi, Then the Basidiomycota are the most commonly recognized. Mushrooms, puffballs, bracket fungus, or shelf fungus, and their spores are called basidiospores. So, the Chytridiomycota contains spores called zoospores. A zoospore is an asexual spore that does have flagella, so it swims, because the Chytridiomycota are aquatic fungi. And we're going to skip those two groups. All right, the chytrids or Chytridiomycota include the simplest fungi. Some are unicellular, others do form branched aseptate hyphae. Most reproduce asexually by producing zoospores. Chytrids play a role in decay and digestion of dead aquatic organisms, but some are parasites. So the one that we're really going to look at, talk about is Batrachochytrium dendrobatitis. And this species of chytrid has decimated over 200 species of frogs. Because you may or may not know this, but frogs have very small lungs, so they have to supplement their breathing with their skin. So when they get a skin infection, they actually slowly suffocate to death. These are some images of, this is an image of a zoospore here. Then the Zygomycota produce their sexual spore is called a zygospore. The main example from this phylum is going to be the black bread mold. Zygospore fungi live off plant and animal remains in the soil or in bakery goods in the pantry. The species I want you to know from the phylum Zygomycota is called Rhizopus stolonifera, or black bread mold. It has 3 kinds of hyphae. It has stolons, which are horizontal hyphae. They're on the surface of the bread. Rhizoids, that are hyphae that grow into the bread. And then sporangiophores are hyphae that grow up above the bread, and they have sporangia on the end that produce spores, and those spores will be released when the sporangium burst. Zygospores are diploid spores produced during sexual reproduction. There are two mating types of hyphae. One is, instead of being male and female, we have plus and minus hyphae. They're chemically attracted to one another. They grow toward each other until they touch. Then they form a gametangium. And when the gametangium merges, the nuclei fuse and you get a diploid zygote. And from that zygote, a wall develops around it. We now call it a zygospore. And then the zygospore is going to form the spores that are released and dispersed in the air. Okay, so this is the life cycle of the black bread mold. I'm not really going to go into it. You can take some time and look through it. Pause this and look through it. But it shows down here in the negative mating type and the positive mating type and how they fuse and form the gametangia, and then the nuclei fuse, and that gametangia now becomes a zygote, which is diploid, forms a wall around it, becomes a zygospore. The zygospore forms spores by meiosis, and those spores are released in the air and land on bread and other types of food and infect it.